All right, so before we get too into anything new today, just quick reminder of stuff we've already talked about in these study sessions. When we talk about taxonomy, classifying things, today the system is phylogenetically based, which means you're put into the groups you're put in based on what two things? Genetics and evolution. And remember that the different categories of groups are called clades. And we start with the biggest group, the domain, and work our way down to the smallest group species. As you go down, these groups include less and less organisms, and they're becoming more and more related. We talked about these phylogenetic trees, that they're basically evolutionary trees of family trees of evolution. And that wherever there's a branch in the tree, what's implied to be there? A common ancestor. So whatever was right here, some of its descendants evolved to be the bacteria, the eubacteria, and some of its descendants evolved to be all these things. Well, whatever was sitting right here, some of its descendants became archaea, and some of its descendants became all the eukaryotes. This diagram is trying to show us that us, as eukaryotes, we're more related to which of the other two groups? Archaea, because we share a common ancestor a little closer down the line. Okay, we also talked a little bit about eubacteria, regular bacteria, true bacteria. They're prokaryotic. Talked about that in class today. They have a cell wall, mostly made out of this stuff, which is what's peptidomi? Part protein. What's glycan mean? Sugar. They don't have any membrane-bound organelles, but they do have ribosomes. Their nucleus is not enclosed. Their DNA is just sort of floating around. It's called the nucleoid. And they also have that capsule or glycocalyx, which is sort of like a mucus coat on the very outside. And we said that protected them from bacteria's two main enemies, which were what? Drying out, or what was the more fancy word we used? Desiccation, drying out. Or it also protected them from what? Immune systems, from organisms they may be living inside. We talked about pili, and how they were sort of like bacterial Velcro. They help bacteria stick to surfaces. But there's some special pili that actually help one bacteria attach to another. And they perform a process called conjugation, where essentially they swap some DNA. And that's sort of a form of what? Sexual reproduction, right? And the pieces of DNA that they swap are little tiny extra loops of DNA called plasmids. And the purpose of doing that is so that they get what? Some genetic variation. Because how do they usually reproduce? Asexually. Prokaryotic or binary fission. And we talked how the advantages of reproducing that way. What's the main advantage of reproducing that way? The asexual way. It's fast. You don't have to waste resources attracting a mate, keeping a mate. Another thing that we talked about that eubacteria have are endospores. And we talked about how that if this was a bacteria, it's got its one little looped chromosome, its one little circular chromosome. If the environment starts going bad, what does the bacteria do? It builds a thick coat around the chromosome. The rest of the cell dies. But that thick coat protects the chromosome. And when the environment gets better, what happens? It grows a new bacteria. That makes some bacteria really hard to kill. And if you're trying to sanitize a surface, you got to be really careful because it takes extreme heat or lots of chemicals to kill them. Another thing that we mentioned were that bacteria do cause some diseases. Everything from strep throat to syphilis to tetanus. We mentioned the tuberculosis, the plague, the bubonic plague. Those are all bacterial diseases. But most bacteria actually have a positive influence on the environment. And their main positive influence is the whole decomposition And related to that, doing what? Putting minerals back into the soil, mineral recyclers. Things like nitrogen, phosphorus, and sulfur get recycled through the environment. We've talked a lot about how a lot of bacteria will do nitrogen fixation. They'll take N2 out of the air, break that triple bind, and convert it into what? Two ions. Ammoniums or nitrates, which are forms of nitrogen that plants and animals can use. And then the last thing we talked about with the bacteria were these terms here. If a bacteria is an aerobe, that means it's what? Uses oxygen to do cell respiration and therefore make ATP, like us. Some bacteria are the opposite of that. The opposite of that are called obligate anaerobes. That means they completely do what kind of respiration? Anaerobic. Means oxygen is actually toxic to them. Can't be around oxygen. Some bacteria are a little bit more flexible. They can do aerobic or anaerobic respiration. That means they can do either one. Facultative anaerobes. We looked at this picture of a normal prokaryote, talked about some of the parts, how that they have a capsule on the very outside, a lot of them do. Just underneath that capsule is the wall, be right in here. And just underneath the wall is what? The membrane. Inside there's cytoplasm. There's one main chromosome, which is a big loop. It's not necessarily a circle, it's a big loop. They may or may not have these extra little loops called plasmids, extra little loops of DNA. But they're all going to have these little dot things, which are what? Ribosomes. Some have flagella, which are involved with what? There's some binary fission going on. Binary fission is similar to what process in us? Mitosis. Now, mitosis is not really division of the cell, it's division of what? The DNA. And then what happens after mitosis? Always. Cytokinesis, which is when what actually divides? The cell. So binary fission is sort of like those two things put together. It divides the DNA and the cell, and it can take as short a period of time as like 20 minutes. It can happen really fast. If bacteria were reproducing as fast as they could, if the environment was perfect, and we had a graph of time versus number of bacteria, what would the growth curve look like for them? It'd be like that. What do we call that kind of growth curve? Say again? Exponential growth. Now, in nature, does it really usually work that way? No. Normally. Why doesn't it usually work that way? What keeps it from doing that? You know, they run out of stuff, right? They either run out of space, they run out of nutrients, they run out of water. Something limits their growth. What does the curve usually actually look like? It usually looks something like that. What do we call that kind of curve? It's more of an S shape. It's called logistic growth. And what do we call that level where they start to even out? Sort of where they reach their maximum population. Thomas used something to word a minute ago. They reach something called the carrying capacity. Which is essentially the maximum number of organisms that the environment can support. Now, have they stopped reproducing when they get up here? No. What's happened? Yeah, the death rate and the birth rate have evened out. And they just got a stable population size. We also talked about the shapes of bacteria. How that they come in usually a spiral type shape. A shape that's sort of capsule shaped called a bacillus. And circular spherical shapes called a coccus or cocci. And then one more thing about regular eubacteria. Some of them can do photosynthesis, and those are called cyanobacteria. For a long time, these things were called what? Wrongly. They were called blue-green algae. Why are they not algae? Why are they bacteria and not algae? You have no membrane-bound nucleus, no membrane-bound organelles. Now, most things that do photosynthesis have chloroplasts, right? Organelles. That's where the chlorophyll and stuff's at. Where's the chlorophyll at in this thing? What is that? That's the membrane. It's in the membrane. That's how it works. All right, another thing that we need to briefly talk about are the archaea. Remember, they're also prokaryotes. And when scientists first found them, they thought they were only found where?
Pennywood, you have a call on line 1, please. Pennywood.
Places that were really salty, really hot, really cold, really dry. But what they found since is that they're found all over the place. What separates them from the regular bacteria are a couple things. And I'm going to go to that slide. First of all, some similarities between them and regular bacteria. These things are all similar between the two. They're both prokaryotic. Neither one of them has an enclosed nucleus or membrane-bound organelles. They both have cell walls. They're similar in size and shape. They're also similar in terms of how they reproduce. But what makes them different are these things over here. Even though they both have cell walls, the archaea don't have that peptidoglycan stuff in their walls. Their walls are pretty diverse. Their ribosomes are different. They both have ribosomes, but they're different structures. The archaea ribosomes are more similar to eukaryotic ones than they are to regular bacteria. And that's one of the reasons that phylogenetic tree that we saw earlier looks something like this. With this first branch over here by itself being what? Regular bacteria, eubacteria. And the second branch off right here would be what? Archaea. And then these would be the eukaryotes over here. So the reason that they're drawn this way is that a lot of the chemical characteristics of archaea are more similar to eukaryotes than they are eubacteria. Another difference, eubacteria can make those endospores that we talked about. Archaea can't, as far as we know. And another big difference is, as far as we know, none of the archaea make people sick. None of them are pathogenic. All the pathogenic bacteria are regular old eubacteria. Make sense there? Right, one other thing that we want to hit today that we haven't talked about at all here. That's a little bit about viruses. Now, we're talking mostly about living things. What about viruses? Not really. Where do viruses go in the classification system? The domain, kingdom, phylum thing. Where do the viruses go in there? They don't. There's a different classification system for viruses because they're not really living according to most definitions. They don't have cells. The name virus actually means poison. I think it comes from Latin. But a good definition of a virus would be this right here. Obligate intracellular parasites. Obligate means where? Means what? Just like obligate, what does it mean? You're obligated to do something, what does that mean? You gotta do it. So this is saying they have to be where? Inside a cell. And once they're in that cell, they're doing bad things to that cell, right? They're benefiting, and the host cell is not benefiting, it's being harmed. We'll talk about a lot of viruses along the way, but one kind of virus is called a bacteriophage. And if you were in one of the classes today, I don't remember which one it was, we talked about the meaning of that root right there, phage. Anybody remember what that meant? I think we were talking about macrophages. Phage means to eat. So literally that word means what? Bacteria eaters. These are viruses that attack bacteria. Bacteria can get sick too. There are viruses that actually attack them. And the reason we talk about bacteriophages is they're pretty simple viruses. Most viruses are made out of two things. Protein coat on the outside, called the capsid, and then some kind of nucleic acid in the middle. And unlike you have DNA and RNA. They have one or the other typically. They either have DNA or they have RNA. Not both. One or the other. We talked the other day about retroviruses. And why are retroviruses called retro? They only have RNA, which scientists think was what? Was the first genetic material. But what's not so good about using RNA as your genetic material? It mutates all the time. Think back to the central dogma of genetics. DNA, mRNA. What's that step called? Transcription. And mRNA proteins, that's called translation. And then the proteins determine your traits. You also got to replicate your DNA. But the retroviruses can't really do this because they don't have what? They don't have DNA. So they have an extra step. And they have to, when they inject their RNA into a host cell, for it to work, that RNA has to get turned into DNA. And what's that process called? DNA to RNA is transcription. RNA to DNA is reverse transcription. There's an enzyme that does that called reverse transcriptase. That process, though, makes a lot of mutations along the way, which makes these kinds of viruses pretty wet, pretty dangerous. They're constantly changing, and our immune system doesn't know how to deal with them. Name me 3 examples. Heard the cold, common cold. I heard Ebola, the influenza, flu virus. One more. Maybe the one you know the most about. HIV. HIV is another retrovirus. All right, something else we want to talk about is called the lytic cycle. And there are those letters L and Y together again. When we see an L and a Y together in this class, at the beginning of a word, what does it typically mean? Like hydrolysis. Split, break, tear. The lytic cycle is the life cycle of a virus. It's the name for the life cycle of a virus. And I've got a picture here that I'll show you why we call it that. Here's the lytic cycle. For a virus to work, and here we're talking about a bacteriophage, the most simple virus. The first thing that happens is that the virus attaches to some kind of receptor on a host cell. Now, does that bacteria, or if this was your cell, do you have a receptor for a virus? Would that make sense? No. So what's that virus doing? It's using a receptor for something else. It's found a way to use receptors for maybe a hormone or something. So it binds to that receptor. And once it gets there, what's the usual signal when something binds to a receptor? What does that usually tell a cell to do? One thing that it often tells cells to do is pull in whatever that is that's attached to the receptor. So it does that. Is that a good idea for that cell? The virus is tricky. So it pulls it, takes it in, and step 2 of this is called penetration. Usually the part that goes in is just the DNA or the RNA. That part will go into the host cell. Step 3, biosynthesis. Once that DNA goes into the host, can that cell tell the difference between the virus's DNA and its own DNA? No. So it does whatever that DNA says to do, which is usually what? make more viruses, which is what's going on here. So it makes the pieces here, and then in step 4, it actually puts those pieces together. Until eventually there's so many viruses, so what happens to the cell? It explodes. And that's why it's called the lytic cycle. We might say the cell lyses, it breaks open. How many viruses are we talking about coming out of that one? Potentially thousands. And each one of those could affect another cell. So that's why the growth curve of the viruses would be extremely exponential. That's the lytic cycle. From attaching to a receptor, injecting the DNA or RNA, building virus components, putting them together, and then bursting out of the cell, killing it. Now another thing I want you to notice is this cycle down here at the bottom. Sometimes the virus will sort of take a time out. This is called the lysogenic cycle. Notice what happened in this case is that the virus DNA got injected, just like it did before. Steps 1 and 2 happened just like before. But that viral DNA actually becomes part of the host cell's DNA. Here's the virus DNA. Here's the host cell DNA. They're together. They become part of each other. And that virus DNA is just sort of sitting there not doing anything. It's sort of like it's in hibernation to some extent. But eventually, what will happen to it? It'll become active again and it'll go back into the lytic cycle. There are a lot of viruses that go through this kind of stage where they sort of seem to disappear for a while. The symptoms disappear. Name me one. A virus that you can have, but the symptoms go away and then they come back again. And then they go away and they come back again. And they go away and they come back again. Herpes is one, like the STD. You have symptoms, symptoms go away. Have symptoms, symptoms go away. You never get rid of it. Once you have herpes, you got herpes. Chickenpox. Once you have the chickenpox virus, you got the chickenpox virus. Later in life, it may come back as shingles in a different place, but it's the same virus. Because that virus DNA is now part of your DNA, and there's really no getting rid of it. It's there. It's just hiding. What will often trigger the transition from lysogenic into lytic? What might cause that to happen? What's going on lots of times when you get shingles or herpes outbreaks of symptoms? Something's going on with your immune system. Maybe you got HIV and your immune system is just like whack. Or maybe you're really stressed out. Maybe you got some kind of cancer. All those things weaken your immune system and cause those viruses that probably wouldn't have popped up to pop up again. So the lytic cycle is the normal way a virus works. The lysogenic cycle is when the virus DNA is incorporated into the host cell DNA, and the symptoms may disappear for a while, and then they'll come back out. And then they'll go back into lysogenic, and then come back out. And that cycle just continues over and over again. While we're here, let's look at a couple of pictures. These are just some pictures of viruses. Everything you see up here in blue is the capsid, which is the code of what? Protein. And then everything you see in red, that's the genetic information, which is either DNA or RNA. This is a bacteriophage. This is one of those viruses that attack bacteria. Sort of looks like a spaceship or something. Pretty cool looking. Here's a tobacco mosaic virus. Can plants get viruses? Apparently they can. That's one that affects tobacco plants. Here's the flu virus. Let me go back a second. Make sure I didn't miss any terms here. So let's go through some examples of diseases caused by viruses. Smallpox, which as far as I know is the only human viral disease that what has happened to. We have eradicated it. When was the last known case of smallpox in the world? Yeah, I'm getting there. When was the last known human case of smallpox? In the 70s. Hasn't been a case since the 70s. How did we eradicate it? How would you get rid of a virus disease? You vaccinate everybody that you can. And eventually, smallpox, the virus, can only live inside people. So if most people have been vaccinated, it goes away. Now, good or bad, for good or bad, our government kept some smallpox virus. It's in Atlanta. It's the Centers for Disease Control. Locked away somewhere. The Russians kept some too, somewhere in Moscow. Supposedly those are the only two sources of smallpox left. Why did we keep it? For political purposes, the reason is to study the diversity of viruses. For real reasons, we probably kept it to develop a biological weapon. And it's the same reason the Russians did. So, one thing that we worried about after 9/11 was maybe these terrorists had gotten a hold of this. Have you guys been vaccinated against smallpox? Stopped sometimes in the 60s vaccinating people in the United States because there hadn't been a case in the United States since 50s maybe. The last cases were in Africa. I haven't even been vaccinated against smallpox. So if somebody let smallpox loose in the United States, your parents probably be okay. Because most of them are probably vaccinated. They were probably born in the 60s. They're probably okay. But people my age and younger, we wouldn't be. So what did we do right after 9/11? What did the government do? They made millions of doses of smallpox vaccine. They didn't give it to anybody. It's stored up in a bunker somewhere so that if a case of smallpox ever shows up, they can quickly start vaccinating people. And hopefully quick enough to keep people from dying. Scary stuff though. Polio. What does polio do to you? Yeah, it causes muscle and nerve problems. It can cause you to become crippled, like have a really bad limp. It can also paralyze what really important muscle? Diaphragm. It can cause serious breathing issues. Yeah. I don't know. As far as I know, we still have a vial. But I may be wrong. So the plague is actually not that rare, though. It's a bacterial thing. It still happens. Yeah, the plague is a bacterial thing. It's still around. You can still get it. It's treatable with antibiotics, though, so it's not as dangerous. Both of these diseases we have had vaccinations for at least 50 years. And those same vaccinations still work. So what does that mean about those two virus diseases? They're DNA viruses. They don't mutate that much. Those are easier to control. AIDS is obviously viral. Influenza, the flu, viral. Chickenpox is viral. Common cold, Ebola. If you get a virus disease, typically if you go to the doctor, what should a good doctor tell you? You got a virus, can't really help you, go home, take a nap, drink plenty of fluids, rest, hopefully your immune system will take care of you. If not, you're in trouble. That's what a good doctor should tell you. Now, there are just recently a couple of drugs that have been developed called antivirals. For example, for the flu, what's the drug? I bet some of you've taken it before. Anybody heard of Tamiflu? That drug is a virus enzyme inhibitor. There are similar drugs for AIDS, right? Magic Johnson, the basketball player. You guys know Magic Johnson, right? How long has Magic Johnson had HIV? 20-something years at least. And he's still doing TV commentary. Hey, he's healthy. because he's taking huge doses of these antiviral medications every day. Notice on what it's costing his insurance company. But you can't control HIV with these antivirals. That's not true for most virus infections, but we do have some diseases, some treatments for some of them. Another couple of things I'll mention, and then I'll call it a day. There are these little particles called viroids, which are, what do we know that means after our discussion today about the letters OID? Sort of like a virus. They're simply pieces of RNA. No protein coat, no capsid. They're just little pieces of RNA. And they cause a lot of different plant diseases. Why not animal diseases? What do we have that plants don't? Immune system. And our immune systems will protect us from those kinds of things. We don't typically get infected by them. I'll mention one thing that's kind of scary, and then I'm going to show you a little video clip and call it a day from there. There are these things called prions. They're infectious proteins. No DNA, no RNA, no replication, transcription, translation. Just pieces of protein that are infectious. And they usually cause really bad disorders of your nervous system. What's the most famous one of these? Mad cow disease, which you get from just eating beef, right? It's really rare, but does cooking the beef help you out here? No. Cook the beef to a crisp, and if you eat it, you can get the mad cow disease. What does it do to you? It essentially destroys your brain. It's like Alzheimer's disease on steroids. Where was the last outbreak of mad cow disease? The last famous outbreak of it? It's probably in the late 90s in Great Britain. It's a big outbreak of it. A lot of people die. If you get it, you die. Essentially, there's no turning back once you get it. It's really rare though, and typically you're going to catch it from eating nervous system parts of a cow. Probably not going to get it from eating steak. But if you were to eat cow brains, yeah, or something close to the spinal cord, you might very likely get it. On the surfaces in your room. Well, I mean, if you cook it, you're going to kill those germs anyway. I guess what they're worried about is getting the bacteria like salmonella on your kitchen countertop and then you not cleaning it off and you touching it and sticking your finger in your mouth and that kind of thing. All right, I'm going to stop my recording there. I'm going to show you guys, before we leave, a little video clip. It's like 6 minutes or something like that. And for you guys who are going to watch this on the recording, I'm going to flash up the URL for people who are watching this online later. Here's the URL where you can find this online.